Brian Kilburn. I'm the Senior Scientist and Services Manager at Arima. I'm going to talk to you today about generating high-quality reference genomes with Arima HiC. Just to give you a little bit of an overview what I'm going to talk to you about, give you a little bit of an intro into who Arima Genomics is, discuss our 3D genome technology overview, briefly talk about the benefits in genome assembly, and then get into what we're going to be doing here for this week's training is talk about our high coverage HiC kit. Just a little bit about Arima. We're based out of San Diego, California. We actually just moved to Carlsbad, which is about 20 miles north of San Diego. Right now we have about 30 plus employees that are on site in Carlsbad with multiple sales and marketing teams spread across the United States and here in Europe as well. We have 700 plus customers and over 300 publications as of today, and that's still counting. What makes Arima HiC kits good or top of the market is the flexible sample input. We have standard and low input requirements. It's designed to work over many different workflows in your lab. We've been commercializing this technology for probably the past 5 years. We offer not just products, we also offer comprehensive services. That means you can send your samples directly to our lab and we can run the HiC directly for you, send you your sequencing results back, your libraries back, whatever you prefer. All right, so let's talk a little bit about the Arima HiC difference. So the main thing is you can take your Arima Genome Assembly HiC solutions right from a draft to a reference genome. And it's very easy, it's very robust. It's probably the most robust HiC technology, especially across a range of different applications. We have end-to-end solutions to go from sample to discovery. It's user-friendly. The workflow, you can really finish it in 6 hours if you want to. There are some stopping points in there if you don't want to work for 6 or 7 hours per day, where you can stop and do a couple overnight incubations. But if you really wanted to push your luck, not really push your luck, but if you really want to work hard that day, you can get it done in about 6 hours. That would get you up to library prep. It's integrated with many different standard library prep protocols for next generation sequencing. Again, what we kind of pride ourselves on is, the sample requirements. We have very low input, works with many different sample types, and you can send us DNA or you can work with DNA from many different organisms. And then we also have world-class service and support from our dedicated scientists. So now let's dive into what the 3D genome workflow looks like. It's powered by HiC, which is high throughput chromatin conformation capture. It reveals how the 3D structure of DNA impacts gene regulation and cellular function. A little bit about how it works. We extract DNA and we preserve the 3D structure by cross-linking, cutting, and religating to the DNA. The process captures the proximity information within the nucleus. After fragmentation and sequencing, we can analyze the data to reveal the 3D interactions within the genome, which provides crucial insights into cell type diversity and the overall genome structure. So how is HiC used in genome assembly? Traditionally, traditional sequencing methods result in hundreds to thousands of unmapped contigs. This lacks the necessary information and context for a complete assembly. HiC technology is critical because it captures the 3D structure of the genome. So with the HiC, we can scaffold these contigs into their correct locations in the genome. And this leads to a more complete chromosome scale assembly, which in turn reveals critical information about the biodiversity, conservation, things like that. And we can even use it for human health. So how does HiC improve genome assemblies? There's 4 main ways. It orders and orients the contigs. It anchors the contigs to chromosomes. It fixes misassemblies and or identifying structure variation. And it's also used for phasing haplotypes. So let's take a look at these a little bit more detail. So first, HiC data helps in ordering and orienting contigs. Current long-read technologies provide high quality contigs, but what happens is they lack the context to arrange them correctly. HiC technology offers DNA proximity information, which is used to order and orient the contigs. For example, researchers were able to organize contigs in 2 lentil genome assemblies, and these were scaffolded by HiC data. Next, HiC data aids in anchoring contigs to chromosomes. As you know, there's highly repetitive elements and pseudochromosomes. They often hinder, scaffolding, which makes it very difficult to complete the chromosomes. HiC data provides long-range contiguity information, which overcomes these challenges, and it provides long-range contiguity information. HiC data is also crucial for fixing misassemblies. As you know, there's many repeats that can result in incorrect scaffolding. So HiC provides chromosome territory information. It also helps correct a lot of the errors that you see. So if you look at this example here, this is a heat map that identified some misjoins in the 34 hummingbird chromosomes. They were manually curated, which made your more accurate genome assembly. Lastly, HiC data is involved in phasing haplotypes. So phasing alleles require insight into chromosomal interactions to cluster contigs correctly. HiC data enables clustering and scaffolding of the individual haplotypes. For example, here's HiC data that was used to cluster in the wild sugarcane genome. So you can see that chromosome 4 was phased fairly well using HiC data. So just to summarize this, HiC data enhances genome assemblies. It provides a necessary context to order and orient contigs. It anchors them to the chromosomes. It fixes misassemblies, and it also phases haplotypes. These improvements lead to way more accurate and complete genome assemblies, and it really just demonstrates the critical role of HiC technology in genomics. So I kind of talked a lot about, the benefits of HiC to genome assembly. So let me introduce, our HiC solution and how it's been designed to optimize assemblies. So what are the issues with HiC? So many researchers, basically, they prepare their own HiC reagents. Making that chemistry or using with what we call is off-the-shelf products. Not really off-the-shelf products, but homebrew methods. What that does is it causes many problems because the chemistry is difficult to build. It can dramatically vary in performance. So when we're talking about how can we make HiC better, a lot of the problems that we saw and the concerns we had was, how do we make this work across different species, different sample types, different inputs, collected under different conditions, and to make it work every single time? Because what was out there before was unreliable chemistry, which can be very, very costly. Our solution to that was Arima HiC, high coverage HiC kit. So I'm going to introduce you to that now. It is basically a superior chemistry for better genomes. Yeah. And what it is now it is a 4 restriction enzyme based chemistry. Our flagship product was basically 2 enzymes. So if you look over here at the first chart, it's just a uniformity plot. It shows normalized sequencing data. And on the x-axis, you see the sequencing coverage. On the y-axis, you see basically the number of bases that were sampled. The second and third charts are just in silico models. They were generated using VGP genome data, basically showing sequence coverage. So what our challenge was is how do we make a high quality, cost-effective chromosome scale assembly with a kit basically right off the shelf, don't have to change anything for sample types, sample inputs. It works just about every single time. So here's a look at our Arima high coverage HiC workflow. It's similar to OmniSeq, similar to others, other than we use a restriction enzyme based assay. So basically, with our high coverage HiC kit, we basically can get way better uniformity, way better coverage, and that's across all sample types, even with standard input and low input samples. So the high coverage kit yields high quality 3D genome data. So let's actually look at the protocol that you guys will be executing during this week of training. Basically, the DNA is cross-linked to capture the proximity relationships within the chromatin. The DNA is in sequence-independent manner at that point. DNA is then ligated to proximally adjacent DNA, which is captured with a biotinylated oligo bridge, and then reverse cross-linked to create molecules ranging in genomic distance. So, what are the critical steps to this procedure? I'm going to give you a couple tips for success. Most of my tips that I'm going to give you have nothing to do with the Arima kit. It's basically the way you're processing your samples. Once you get to samples that have as much DNA as you can possibly get, most of our kits work between 500 nanograms, or they've been optimized for 500 nanograms up to 5 micrograms, but we have solutions for low input as well. The main thing is determining whether your samples are standard versus low input. As I said before, we can work with many different sample types. So for cells, our standard input says 5 to 10 million cells. Low input is less than 1 million. We actually have some data showing that the high coverage HiC kit work with less than 50,000 cells. For large animal tissues, which I'm not sure if anybody here will be working with large animal tissues, our standard input is 50 to 200 milligrams. Low input is less than 50 milligrams. It will work with much less than that. The big thing with low input samples is making sure we, as a group in this lab, ensure that we don't lose anything during our processing, during our pulverizations, our wash steps, our cross-linking steps. That's where you're going to have the most loss. And if you don't have enough input going into your HiC, you're not going to have enough yield. But it's not usually because of the kit. It's usually because we have lost so much in our processing and our washes and our steps going forward. So just because I talked about the protocol, and we'll talk a lot more about the protocol during the training. I had a few slides earlier that I kind of took out because I think there are things that are going to be, everybody's going to have different questions in different areas that they want to try and optimize. There's very little optimization that needs to be done with our kit. But I just wanted to mention that some of the largest genome sequencing projects, like the Vertebrate Genomes Project, the European Reference Genome Atlas, and the Sanger Institute, with the Darwin Tree of Life, they have all took our kits and have been using them for their publications and for their labs, and that they ensure that they get the best genome assemblies quickly and every single time. We now have over 200 genome assembly publications, and that's still counting. I think there's 4 or 5 that are in progress. So genomes are a strong foundation for diverse areas of research. The Arima high coverage HiC kits and even our HiC kits are utilized in agriculture, sex chromosomes, conservation, translational research, and even in human genomics. So just to kind of conclude what I kind of talked about, you can't get phased chromosome scale genome assemblies with sequencing alone because you're missing the sequencing context, which is required for orienting and ordering the contigs, correcting scaffolding errors caused by long repeats, anchoring contigs to chromosomes, and for phasing haplotypes. So what's the solution? It is our high coverage HiC, which addresses all of these issues. It provides enhanced uniformity through 4 enzyme chemistry. It's consistent and it performs very well across many different samples, different species, and different inputs. You get high quality 3D genome data, and you get more usable high quality reads. So thank you.
So we can be confident if it's less than 50%, we will know that maybe we don't have.
I mean, passing is right around 15%. So if you're at 50%, that's actually really good.
Yeah, 15, yeah.
Yeah.
But if it's very high.
No, we see it sometimes as high as 75%. It's not out of the ordinary, but those are very, very good samples. You know, they're not lichens or something that's been kind of sitting around for years and years. You know, these are high quality cell lines that we see perform really well. So it really, it's going to depend on your sample input at the beginning. How good is your sample at the start? Yeah. But you're right. And like I said in the presentation, I think the biggest pitfall or the biggest thing for you guys as trainees learning this and doing this is your sample input. You know, you want to start off with high quality samples. You want to get them cross-linked as quickly as possible. That's where most of the mistakes are made. It's not usually when you start with the lysis. It's by that point, it's either going to work or it's not going to work. And it's what's upstream, and it's your sample processing that is the most critical for this assay.
Actually, it also depends on the organisms.
True, and that's true, yeah.
Because for
Different organisms.
it's developing more than 15% for the QC1. It's less than 50%, but we continue and the library are okay.
And I would suggest that even if your QC1 says it failed, go on to library prep, see if you make libraries, do shallow sequencing, because it may still work. And I agree with you, the complexity of libraries is an issue. And that's when you start getting into low input samples, the complexity is, and I think Miles and you guys will agree too. When you have low input samples, really low, yeah, you're going to get something, but it's not going to be as complex as if you have standard input or a lot more sample to work with. So it's kind of the nature of the beast. Yeah. Yeah. And I suggest, you know, and as everybody probably knows, always use fresh formaldehyde. You know, don't use old formaldehyde. We get calls all the time where, oh yeah, we cross-linked them and, you know, our data looks terrible. Well, how old is your formaldehyde? Oh, well, I think we bought it like in 2021. You know, it's 3 years old, you know, and then they expect it to work. Or they'll say, well, we made a 2% formaldehyde solution, you know, 3 weeks ago. So we just cross-linked with that. That's not going to work. So it depends on if you're using methanol based or not. I always buy small vials and I use it for maybe a month and that's it. I would make my 2% fresh every single day.
So in all of us, when we were working with the HiC data, we've seen that for some organisms, Arima works best, for some protocols, Dovetail works best. So that is why we'll be trying both on the same kind of samples, just for the giggles, and then to compare the data. But also it seems that the assembly link with their base agnostic approach and yours with 4 different restriction enzymes, which is a huge upgrade from the 2 which we were using for vertebrate genome project. That's really interesting to see because it seems like your uniformity and the coverage across the genome has increased dramatically.
It has.
So looking forward to analyzing the data. So
Sanger has used the 4 enzyme chemistry quite a bit lately. And they were actually one of the groups that helped us design this kit. They were kind of our protocol based
rabbits.
Yes, exactly. They beta tested it for us over a lot, and they came in with their suggestions, and it works very well. It's a pretty robust kit.